Okay, well, I thank you for letting me speak today. I'm going to be talking primarily about my response to the discussions yesterday. And I wanted to first list some of the people who worked on these projects over the years and highlight some of the people, many of whom are in the room today, who are particularly active in the things I want to talk about. I'm going to be focusing on multi-scale multicellular modeling, or virtual tissues. And I know a lot of people know this as well as I do in the room, but there are some people coming from different backgrounds. So I'll give a little bit of background about that. And then I'm going to talk a little bit about some of the challenges that we face if we try to standardize in this domain. And then I may make some suggestions about possibilities for approaches. So we're in the context of organismal organization, development, function, and failure. And we're in a world where we have to consider both biochemistry and biology and physics interacting with each other. And so trying to understand the dynamics of these very complex systems requires not only a modeling of the biochemical networks, but of biophysical phenomena as well. The classic sort of virtual tissue representation that many of us work with takes a 3 or 4 level approach. It starts with cells as the active agents in the middle. It typically includes some kind of network models for control inside of cells. It puts those agents into a spatial context, particularly looking at diffusible chemical fields, but also potentially looking at the mechanical context of cells. And then often those are put inside of a non-spatial model, for example, a PBPK model of a transport in the whole body context. And so we have at least 3 levels of modeling that we have to think about. There are a couple of fundamental characteristics of this kind of modeling, some of which are shared with network models. These are mechanistic agent-based models. They're multi-scale. The focus is on emergent phenomena resulting from interactions between agents. They're mechanism-driven. They're additive. We don't get anything for free in these models. We typically are moving from cell to organism level, although certainly these modeling approaches can be used to model what's going on inside of individual cells. And they're based on quantitative submodels of object behaviors in particular, what cells do, what other components do. And their outputs are typically movies. And that differs from a network model where you typically have time series of scalar values. If we can get these to work in an effective way, there is a lot we can do in terms of scientific discovery, in terms of biomedicine, in terms of engineering. Many things that we would like to be able to do with this kind of model. But till now, they have not been applied at the level that one might hope, perhaps. These kinds of models have been used for more than 30 years. And a big part of the problem with this kind of modeling is that both the methodologies and the models themselves have typically been one-offs. And so reusability and progressive improvement of models has not been something that has been a primary focus. Building useful virtual tissue model these days from scratch really takes about a year for a good postdoc, and it requires people who have a very broad range of expertise that's not so easily found. You need people who understand biology in a pretty deep way, in a variety of scales. You need to have somebody who has some pretty decent physics knowledge, computing and mathematical biology knowledge, and physics knowledge. It's not easy to find people who can do all of that at once. I've been showing this slide now since 2009. We still haven't made it there. I've had a dream for a long time that you would be able to take an experimental image, In this case, this is a famous experimental movie from 2008 of the cells in a developing zebrafish, and would be able to annotate that movie and make a simulation by annotation of an experimental image that could then run and register with the experiment. I think it's worth asking the question, what would it take to be able to deliver that? These days, cell atlases and single-cell omics are great developing technology, but those involve fixed tissue. We really want to understand the dynamics of living organisms. And so, again, if we could annotate fixed tissue experimental data sets and then turn those into living computer simulations, that would make them into much more valuable predictive dynamic tools. The other thing that we would like to do is to be able to enable knowledge generation workflows. And we can imagine what a workflow involving virtual tissues would look like. How you go from experimental data to models and from models back to understanding requires a relatively well understood in certain ways, but nevertheless complex cycle with a lot of steps. You have to be able to build models effectively. You have to be able to validate them. You have to have approaches to running simulations in a controlled way, organized ways of presenting the data that come out of them, analyzing them, doing optimization and validation, and then pulling that data back and integrating it for knowledge extraction. In this case, I have CompuCell 3D listed, but this could be replaced by any of the many modeling frameworks that have developed, Morpheus, PhysiCell, TissueForge, and so on. We don't have the kind of integration tools really at this point. I think Morpheus is working on this, of course, and also PhysiCell, but we don't have as many integration tools as you would have in the network modeling environment, certainly not anywhere near the number of tools that you have in the context of bioinformatics and omics. If we want to be able to realize any of these visions, we have to be able to build complex predictive virtual tissue models grounded in experiment, and we need to be able to build them quickly, reliably, and reproducibly. And that means we need standards. And when we think about standardization, it's pretty challenging. If we look at that workflow, we see that there are a number of different, fundamentally different kinds of questions that we have to address. And all of them are important, all of them are challenging. But I think it's important to call out that there are a variety of different problems that need to be handled if we're going to be able to deliver this. It's not that we don't get value from addressing any one of these individually. Each one has immediate practical payoff if you can do it. But to deliver that whole workflow, you need to do all of them. One of them is trying to build software interface and capability standards so that you can interconnect software components. At the moment, if a beautiful PDE solver is written for PhysiCell, we can't reuse it in CompuCell. A beautiful extension for capability that's written for Morpheus can't be pulled into Tissue Simulation Toolkit, and so on. And so the ability for software components to interoperate and for software frameworks to communicate, I think, is something that would have great value. And I think there's a lot understood about the computer engineering necessary to make that happen. Model specification standards are a separate issue, and that would be particularly to enable portability of models between software frameworks for validation and reuse. And again, this is something that Morpheus has begun to work on. Paul Macklin and Multicell DS have tried to work on aspects of this. I want to make sure that I emphasize that this is rather different from the question of, can we build specifications that allow interoperable model components? so that we can build simulations from higher level components than we do now. I think that the second one, they're both quite difficult for a variety of reasons that I'll address. But the second one, I think, is really the critical thing to enable that dream of building automated model construction. And I think it's quite challenging in a variety of ways. By the way, you could have one without the other. You can have a model specification that specifies an entire virtual tissue that could run on multiple platforms, but isn't componentizable, so you can pull pieces out and reuse them. Similarly, you could have interoperable model components that only work in one framework, only work in PhysiCell, and are not portable between frameworks. So they really are addressing, in a sense, not completely orthogonal, but different problems. Something that I think has not been thought about, but of course people like the SEDML are working on in the context of networks, is better ways of describing the capabilities of real wet lab experiments and also computational experiments, and bring those together so that you can actually build simulations that address what's possible in the real world, but also consistent ways of describing optimization, validation, parameter identification, and so on. It would be helpful to be able to have descriptions of the workflows themselves that involve all of these components. And of course, data archiving and annotation standards for model and experimental data reuse. Again, time series are simpler than the kinds of movies that are generated by these kinds of simulations. And how we deal with movies is still something that's really pretty poorly developed. If I start opening up each one of those headings, I'll come up with a lot of subheadings. I'm not going to go through this laundry list, and I'm sure that people in the room could suggest many more and correct many more. And I'm really bringing these issues up not only not as an expert, but rather as a set of talking points, asking for the advice of people, especially from the SPML community, but also from others who've had experience and success in grappling with many of the problems that we're just beginning to address here in the context of virtual tissues. TJ, I think, will be talking about, and talked about a little bit yesterday, his efforts to build modular interconnect standards for software components. And Iran has worked on this quite a bit as well. Something that would be quite interesting, that's a little bit more challenging perhaps, would be addressing how you can make the software work across different platforms. How do you make the same software work on GPUs? How do you make it decomposable or parallelizable? How do we do software validation? There are groups in Europe that have tried to do white boxing, where you include validation tests within software modules. I think that's a very interesting question. Are there ways we can actually specify the capabilities of frameworks themselves so that we have things like the standards level certifications in SPML? I think that would be very important. I think when we come to model specification standards, which really is where I've focused my thinking, and especially the idea of building software model components, I think then there are, again, many different questions. Whether we specify processes in a shareable way, we specify initial conditions in a shareable way, whether we develop graphical tools to be able to represent complex models in a way that's more intelligible. Network diagrams are very powerful, a way of communicating information intelligibly. We don't have equivalents for the complex concepts that exist in virtual tissues. And so these are, again, drilling out of these many, many headings and thinking about some of the components that would need to be developed in terms of modeling framework standardization. I want to emphasize, and I'll come back to this, that for many of these cases, you don't have a single standard. You have to at least have 3 levels of abstraction, and the solutions at different levels of abstraction may be different. People have different names for these, but I would call them conceptual models that address essentially model structure primarily. quantitative models that are method independent, and then computational models that include methodologies, computational methodologies, specific instantiations. And I think you always are going to need at least those three levels of abstraction when you deal with these problems. If I want to come back and focus on one of these areas, I particularly am attracted to the question of what it would take to build a library of reusable, instantiable biological model components that we could use, example, to turn an experimental image into an executable model. And this is not easy to do. And there are challenges in a variety of directions. The biology, the physics, the mathematics, and the computing all have challenges that we need to address. The biological reality doesn't make life easy for us. Many of the problems that we have to face here are not ones that SBML really, at least core SBML, has addressed. In particular, core SBML really doesn't try to think about interconnecting models that were built independently. If I have two SBML models that involve P53, I can't assume I can go to biomodels, pull out two P53 models, and that they will automatically talk to each other and work together. That's something that's much more challenging to do, but it's what we would like to be able to do in this kind of simulation, basically because we have lots of individual components that we need to be able to interact. Of course, we know that there are many scales in biology and that they interact in complex ways. Something that is quite important that we don't face so much in network models is that there's a lot of reorganization going on. And so the network, if we think of the interactions between cells, for example, in an organism as a network of interactions, that network is continually remodeling itself. And we know that modeling networks that are remodeling dynamics of networks rather than dynamics on networks is a pretty hard problem. Another thing that we have to face is that these are intrinsically phenomenological models. We can't regress to modeling everything in infinite molecular detail, and that means that we're continually making choices about granularity. And so the way we describe the same object will depend on the type of problem addressed. That makes shareability much more difficult. We have many numerical methods at different scales, and within scales, we have to make them work together. We don't have unidirectional flow of information, and that means that software architecture and software coupling to make efficient simulations is more challenging. Again, we can think about the methodologies. We tend to focus on the fact that we specify things like cells using different methodologies in a platform like PhysiCell and CompuCell. And that certainly is a challenge, but I'm not sure it's the primary challenge here. I think that there are more fundamental conceptual challenges about the way you organize information that are as or more difficult than whether or not you're on a lattice, for example. There are a growing number of virtual tissue environments that try to simplify model construction and execution. We have lots of people here from Morpheus. We have people here from Simune. I'm not sure we have any chaste people here. I don't think Roland is here for Tissue Simulation Toolkit. We have people from PhysiCell. TJ will be talking about Tissue Forge, and there are many, many other packages. These differ not only in their methodologies, but in the range of concepts and structures they support. And something that we found a sobering learning experience was trying to exchange models between Morpheus and CompuCell. Morpheus and CompuCell use a lot of the same approaches, and it's still quite difficult to build models that are transportable between the two. We have been developing for a long time CompuCell. It's typical in the sense that it has a model construction tool, it has a model execution tool, it supports those three levels, 3 or 4 levels of different kinds of model structure. Like a growing number of tools these days, It does allow network modeling with SBML specified models, or in this case also stochastic Boolean models with MabBoss. And that is not a bad place to be. But one of the things that we face when we're dealing with these kinds of projects is that there are a huge variety of different applications. We don't really know how people are going to use this software. And so we have to be very flexible in our approaches. These are all CompuCell models, but if you go to the Morpheus site, you'll see an equally diverse zoology of models built around Morpheus, and for PhysiCell, the same thing, and for the other packages, the same thing. And so we have to build simulations tools that are maximally flexible. And one of the great difficulties we face is that we really don't know how people are going to use the tools. Here are more simulations. As you see, some are in 2D, some are in 3D. Sometimes cells are unitary objects. Sometimes cells are divided into compartments. The nature of those compartments varies. The level of granularity of the simulation varies. Whether there are additional biomechanical structures varies. The detail of the extracellular environment varies. All of these things have to be supported in a consistent way, and anticipating all of those uses is quite challenging. A couple of definitions. People always see this slide in almost every talk I give. I like to think about models as decomposable into a model structure. In the case of a network, well, that would be the network architecture, possibly including also the reaction rules that are associated with it, and then a set of parameters, which would be rate constants, initial values, and so on, and then the idea of a model output. It's important, I think, to think about what a simulation does or a simulator does, and I don't think I have a good terminology. I'd be open to hearing what people suggest about terminology, but a simulator fundamentally takes the state of a model and time steps it to give us the state at a later time. And to do that seems very straightforward, and we do it all the time, but actually it makes model specification quite challenging. We have to distinguish when we do time stepping between what I would call dynamics solvers or dynamics handlers, which do continuous evolution in time, and event watchers, which handle temporally discrete events. Those fundamentally have different structures. We have these three or more layers of abstraction we have to deal with: the conceptual model, which we hope will be close to the biology, a mathematical or physical model, which should be method independent, but should be conceptually unambiguous in terms of its dynamics, and then the simulation, which will be tied to the particular approach that we're going to use to do the numerical solution. We tend to lose information as we go down this stack. We have to add certain kinds of information, but we lose a lot of generality and what I would call knowledge when we go down the stack. When we think about models, we always have to analyze them. We have objects in the model, the things we're modeling, and they have properties and states. We have their behaviors and interactions. We have the initial and boundary conditions of the problem, and we have dynamics and events. And my thinking about exactly what behaviors and interactions doing, which are processes, has changed a bit over time. And so perhaps some of my slides may not be wholly consistent about this. If we look at these kinds of virtual tissue models, almost all of them include chemical fields, which have certain kinds of similar properties, namely concentrations, primarily as a function of space, diffusion constants, decay constants. They have behavior of diffusion decay, possibly advection, which is a harder problem. Their dynamics would obey the diffusion equation. Initial conditions would be values everywhere. We have to think about boundary conditions. Cells typically are active agents, although we often will jump in and subdivide our cells into subcomponents. Dynamic networks, which are the most well understood component here. And then we have additional structures like solids and links that have to be dealt with. Biology is full of apparently modular concepts: individuals, organs, tissues, cells, molecules. apparently well-defined behaviors, cell division, cell growth, circulation, uh, named interactions. And we'd like to be able to build modular equivalents of these that allow us to combine components and make them reusable and extensible so that we can build libraries. However, when we dig in a little bit more, we find that those biological modularity isn't as sharp as as we think it is. In particular, and I'll come back to this, boundaries are very fuzzy in biology, and identity is very fuzzy in biology, and we're not able to live with that in a computational context. We have to disambiguate those fuzziness, and that's quite challenging. Object identity and processes change when we change model granularity and methodology, and that's a challenge as well. So let me now come to some comments and ideas about what this means. I'm certainly not going to have time to go through the whole list that I made. And a lot of these things may be obvious to certain people. Again, I'm looking for feedback and suggestions as we go forward in this area. One problem is that cells are complex and diverse. They have enormous ranges of cell shapes. It's really not going to be practical to define, for example, a Purkinje neuron as if it were a point-like particle cell behaving as a single individual. It's very hard to define a universal at modeling abstraction for cells because of their diversity. And of course, cells have incredibly complex substructures which affect how they're going to behave. We don't want to fall down the rabbit hole of having to model everything. And so deciding what we're going to leave out is quite difficult when we come to cells. In particular, there's a tendency, if you're doing center model in particular, to think of cells as individual agents. And in many cases, cells behave like individual agents. On the other hand, there are also situations in which subregions of cells act as if they were independent agents. And so the discretization of agents and cells is going to vary depending on the problem that you're addressing. Force generation and movement of and within cells can be very complex. Cell surfaces, and the V-cell people have worked on this for many years in a beautiful way, are very complex and change dynamically. Transport and force transduction across cell boundaries are complex and depend on a lot of macromolecular detail, which typically is left out of our simulations. And again, the choice of granularity will differ for the same object type, depending on the problem being addressed. That's on the biological side. On the computational side, we have different methodologies to implement the concept, for example, of a cell. We can have a cellular Potts model approach. We can have finite element approaches. We can have vertex model approaches. We can have higher, coarser grained cellular automaton approaches. Each one of these has its own sets of, we can have center model approaches which treat cells as point particles or clouds of point particles. All of these have different descriptions of what cells are. Does the cell have a volume? Does it have a surface? Is that surface instantiated or not? We even have broader level questions. Are we going to treat a tissue as a continuum where we have a field, which is a density of cells, or do we identify individual cell agents? Well, if we're talking about a multi-cell model, our cells are probably going to be agent-like. But what about a virus? If we have a concentration of virus, do we treat the virus as individual particles, in which case we have a lot of them, or do we have them be a field? We yesterday heard about modeling of bacteria inside of cells. Do we model those as spatially explicit objects, as agents, or do we say that there's some number or density of bacteria inside of individual cells that are modeled as agents? Being able to describe things in a method-independent way is pretty difficult. We'd like to be able to at least have some basic method-independent descriptions, though, and that we can work with. And again, we come back to that hierarchy of abstraction that I think is always important to address. Structured materials are hard. Fiber networks in particular, we don't really know the physics of fiber networks. And so trying to address that, I think, is going to be very tough. It's a place where I think it's probably better to kick it off for a bit and not really focus on it. But it does come back to bite us a little bit because the biophysics and biomechanics of materials depend on concepts like constitutive relations. And those constitutive relations in physics, we're used to having things like a viscosity for a fluid, a Young's modulus for a solid. Those concepts actually don't work very well for structured materials. If we try to measure, say, the viscosity in a tissue, we discover that depending on the length scale at which we try to measure it, we get totally different answers. We try to measure something like a Young's modulus. The way we measure it experimentally can change our results by many orders of magnitude. Our materials can type switch as well, and how this happens is poorly understood. And so we are faced with a lot of scientific uncertainty in this case about biomaterials that we really can't deal with. In the case of standards, we have to accept it. Homogenization then becomes quite difficult because we don't know what parameters we need to expose from one scale to another. Physics is much simpler in this way than biology. If I come back to the issue of libraries and object privilege specifications, I see I'm going to run out of time before I get to a lot of these problems. I really want to come back to this idea, it's my dream, of having a library of model component templates. For example, cell types, extracellular matrices, chemical fields. These need to be independent and intercombinable. In an experiment, if I bring two cells together, they'll do something. In a computation, if I bring two computational objects together, we're developed independently, and they have incompatible expectations about how to interact with each other and their environment, the simulation will crash. And that means I need to pre-specify all of the behaviors and processes that my object can participate in. And that's tricky because I can't necessarily anticipate how objects will be used. That means one thing that's very important is that whatever I do, I have to have very strong defaults. Defaults don't have, not only for parameter values, but for context and situations where if I put my object into a context that I didn't expect, it doesn't crash. I think robotics, self-driving cars can probably teach us a lot about this. Processes are a pain. Processes play out in time. However, if I think of an object specification as a state which exists at an instant in time, it's the dynamics and the events, the handlers, that have those processes play out. And I can't necessarily tell from looking at the internal state of an object what processes it's participating in. This makes life much more difficult. If I want to do declarative model specification, interactions as processes are particularly difficult because they're contingent. And I think we need to spend a lot of time thinking about how to specify potential interactions, capacities or capabilities, and rules. When does an interaction activate? How does it activate? Can we specify and agree upon between frameworks on approaches to exposing the choices that are made for when things happen, how they happen? There's also a problem that we assume that interactions are separable. You run into this also in network modeling. You have a network where a node is activated by A, inhibited by B, activated by C. It's not obvious how you combine those signals together. That signal integration is something that's very specific. It may not be separable in an easy way. As a simple example, one of the most important things cells do is stick to each other. Well, whether a cell has an adhesion molecule on its surface or not is a property of the cell. That's an object property, it's clear. On the other hand, whether those adhesion molecules are generating a force on the cell depends on whether the cell is in a spatial relationship with other cells. That means we have to define a rule for when that interaction occurs, for example, an interaction range. In reality, there's a lot of additional complication biologically. the cells will reorganize their membranes. The adhesion between the cells will change depending on how long the cells have been in contact. We'd like to be able to define also whether there are local differences. The adhesivity of the cell depends on the specific position in the cell, regional differences, or whether we have global adhesivity. And maybe that's a good use case to start with. We need to think about what happens if we have incompatible interaction declarations between objects. If we're pulling them from a library, it's possible that somebody will have defined how cell adhesion works on one cell in one way, and somebody else will have defined it in another way. That makes it quite difficult. We need a lot more to understand how spatial relationships work. We don't really have language to describe things like adjacency and interaction range, volume exclusion, partial volume exclusion. These are going to differ depending on some of the implementation choices as well. They're not purely biological reality. In reality, we have volume exclusion of spatial components, but very often in simulations, we don't enforce that. It makes it easier to deal with. The last point, I'm going to run out of time, so I want to try to wrap up, and I'll skip some of these things. But there is something that I did want to talk about, which is who owns dynamics? Who's responsible for implementing or specifying how time evolution occurs? And typically in the simulation environments that we deal with, this is some global property of the simulator. The simulator solves the diffusion equation. The simulator solves the mechanical update rules for cell positions or boundaries of cells. And one of the things that's complicated about this from an object privileged model specification perspective is that the dynamics that are going to play out how the processes, what they do in time, don't belong to the object. And so the one core principle needs to be that, if you like, your modeling universe is an object in itself. And one could have a principle which is that you can, because we're talking about interactions, how the properties of an object evolve in time can't be decided by introspecting the object, because what happens to that object is going to depend not only on its own state, but also on the environment, the other objects that surround it, and those are going to change in time. And so an object needs to have within it a set of dynamics and event handlers that handle not its own evolution, but rather the evolution of the things that it contains. And if we have that as a general principle, then I think a lot of the things that we're talking about as difficult become simpler. However, I don't know of any package that really handles that. Networks tend to be dealt with that way. If we use something like libRoadrunner inside of individual cells, we're fundamentally saying that the cell owns a event and a dynamics specification evolver, a handler that operates not outside of the cell or on the cell itself, but rather on models that are contained within the cell. And I think that this is really a very fundamental concept that needs to be explored more. And that's something that I'd like to talk about more. When I use the word universe, it's really any containing object. And I think that that's something that maybe hasn't been discussed. I'm sure that in other contexts where it's been thought about a lot, but in our world, it hasn't been talked about as much. Another problem that we face is this issue of biologically fuzzy boundaries. There's some simple computational problems that are not so simple. Cells have membranes. Membranes belong to the cell. Computationally, when we have two cells touch, usually we have a shared interface between them, and we lose the idea of who owns what. And that winds up being actually a pretty big problem. Where forces act is another problem. And so I think, again, in the interest of time, I'm already out of my time. I'm going to skip my laundry list, but I'll make the slide deck available, and we can come back and address some of these things if we like. But there are a couple of other problems. One of them I want to mention here is that if we're starting from snapshots and we want to do time evolution from snapshots, things like movement become quite problematic. If we see a cell in one position at time 1 and another position at time 2, we know, for example, where the center of mass moved. We may know that how the shape changed. But if we want to understand advection, we then need to know not only how the overall object moved, but how its subcomponents moved, and that's usually not observable. Identity and existential processes are tricky. Computationally, we can't have ambiguity. If we label a cell type 1 or type 2, we have a Boolean choice. If we have one cell object or two cell objects, that's a Boolean choice. Biology is full of partial or incomplete identity change. Two cells start to divide, but they don't separate completely. Computationally, we have to decide, now we had one object, now we have two. Biology doesn't necessarily make that choice. And so we have to define fiat categories that don't correspond very well to that biological fuzziness. I think that there are a lot of things we can do right away. Some of them are very simple, like have unit specifications. Agree that information only crosses between object boundaries through defined interfaces. It would be nice to be able to have languages that describe the capability of modeling software components and frameworks to make them introspectable. There are a bunch of issues about how you implement software components and make them connect that I think are pretty subtle and difficult. I do think that this issue that I want to come back to, which is that objects should also contain the dynamic specifier, the handlers, dynamic and event handlers, not for themselves, but for their things that they contain, is something that we could address more. That hasn't really been thought about, perhaps as much as it could be. Who's responsible for what? What kinds of flags and signals are passed between scales? Again, this idea of strong defaults, not only for parameters, but also for situations. And I think that we should start at the level of conceptual model specification for object classes. I think it's easier to agree upon model structures than for details of parameters. As Herbert always likes to point out, standards are only useful when they're tools to implement them. And so we need tools for model specification, for integration, archiving. Yesterday, there was a good point that it would be nice to have some high-level minimal standards for what's required when you publish VT models, essentially templates for how to present these complex models in publications. And I think that something that we do have to address as we move between abstraction levels is how to reorganize model descriptions from models and parameterizations that are biologically natural to ones that are computable. This issue, for example, of identity and boundary crossing, it's very natural to have fuzzy boundaries in biological and conceptual models. We can't have that when we get to the computational level. How do we do this kind of transition in a way that's not too traumatic and not too complicated? I think SBML does this very nicely. You have a core concept of a chemical reaction. That's a very natural concept for an experimentalist. Then you have that translation of that into, for example, ordinary differential equation, a rate equation, or a stochastic differential equation, which are natural from the computational mathematical point of view. That complexity tends to be hidden. SBML does a great job of hiding that complexity from the user. It would be nice to have some use cases to explore. There are a lot of possibilities there. We can address all sorts of different things in those challenges. We worked years ago, really a long time ago, trying to build this high-level description of cell behaviors. It got stuck. We'd love to try to get it going again at some point. And so I want to conclude with a little bit of a what next. If we want to implement this, it's a big project. It's really a big project. But there are some big new funding initiatives, both in the US and Europe, which might support initiatives in this area. The NIH has a new Common Fund project, which might support some of this. The European Current Horizon framework programs address a lot of this, mainly in the context of biomedical digital twins, which are not the same problem, but have to address a lot of the same issues. We need to prioritize and start. Again, I think initial condition specification is easier because it doesn't have the concept of time evolution in it. It makes it a little bit easier. We could try to agree on dynamics and event handler architectures and interfaces. We could try to build conceptual descriptions of cell types. Chemical fields wind up being a lot easier if we don't include advection. Everybody seems to agree pretty well on how they work. It would be nice to have some standards for describing the capabilities of the virtual tissue frameworks and their components, and for specifying the interfaces of those software components. I think those are all possible. As always, it's hard to motivate the community to focus on interoperability, partly because the problems are so many, partly because the way the community works doesn't really reward that. And I wanted to say we just got, a week or two ago, a small NSF grant from a program called POSE, which is focused on promoting open source ecosystems. And our topic is open VT ecosystems. It's not a big grant. There are not a lot of people on it, but 3 people at this conference, Kyle Sterling, Michael Getz, who are in the room, I think, and Randy Highland, who's been online, are funded by this. And so anybody who's working in any of these areas should be pulling us aside and telling us what you need, because our remit from this grant is to try to start solving the problems that I've been addressing here. And I'd like to acknowledge some of the other support we've had currently and in the recent past to do this work. Thank you very much for being so patient, and I'd be happy to talk with people about your ideas and suggestions. I realize I'm running into your coffee break a little bit. I don't want to keep people from your coffee too much. Thanks very much.
So we are unmuted. Okay, James, can you hear us?
I can hear you fine.
Yeah, okay, so please open for questions.
Hi, James. Thank you. That was an amazing and I think a very important talk you just gave. Please do share the slide deck that when I see it's recording to make sure that this is public. You made a map of just the space that it's a big ecosystem that's required. Many components that are very interdependent on each other. The model components, no model component on its own has much value. The software tools on their own, I could see it would be very hard to fund any individual. Like we go to one of your slides, and it's an important part of the overall ecosystem, but probably not on its own fundable or even scientifically valuable. It's really the whole thing that's required, that's a challenge. And how do we build something like that, especially with an academic system that you need standalone projects. And I think that's kind of part of the challenge. You did a great job of giving the full space. I think people see little bits and problems, and they attack. They take it on and make their own little universe around a piece of the problem. But we can't. Really everything has to come together. So one way forward is to find a kernel that we can all focus on and build up from there. I don't think this is going to self-organize with independent components. It has to start somewhere and build up, and the complexity has to increase. We can maybe even look at biology. You said this open source ecosystem. It really is an ecosystem. We should look at how biological ecosystems develop. Can we I mean, where can we? I mean, what's that?
They evolved messily.
Yeah, it's messily. I mean, but I mean, we can't do it. Yeah, so maybe we can't do it messily because of the problem that you described, which is that we can't put two different virtual cells in the same and expect them to communicate in the way that biology can do that. So what's the kernel like? Where can we start? And what's a small piece that has standalone value that we can all build up from and expect to to continue to bear fruit as we work our way out. Do you have any ideas about like where's somewhere we can agree on to start? What's like the first kernel? I'd love to hear your thoughts on that.
Well, I hope that's addressed more to the room than to me personally. I agree. I agree completely that it's a bit intimidating. I suppose if we were lucky, you could go to the Chan Zuckerberg or Allen Institute and say, do the whole thing. But I know the Virtual Human Embryo Project in Canada has pretty ambitious goals for this. So you can always dream that something good happens. After all, in Connecticut, you've had a national center. Herbert has his center. Although I think even those centers are not really on the scale that would be needed to realize this whole project. It's a pretty big project. But I do think that there are a lot of these things that are individually of immediate practical value. You have graphical representations of models. That's of immediate practical value. If you can exchange model components or do interoperability of software components, even between a few frameworks, that's of immediate practical value. And so I think there are many places where you don't have to do all of it for it to be valuable. Even delivering the pieces individually could be of value. One of the reasons I separated the question of can we share model specifications for virtual tissues between platforms, and can we build virtual tissues from interconnectable subcomponents, is because I think that actually the sharing between platforms is, at least in a limited sense, easier. I think it wouldn't be that hard to write a language that could run on both CompuCell and Morpheus, for example. They're both derived from very similar sets of concepts, but it doesn't solve the problem of model component reuse. And so I think that there are places that we can address. But I'm with you. I really would rather hear the expertise in the room. We have people who've worked on these issues in a variety of contexts for many, many years and very successfully. And so the lessons from the people who've had success, I think, are critical here.
Yeah. John?
Yeah, Ron, I think you're being humble or something, because in, the Herbert Center, the Center for Reproducibility, the renewal proposal specifically talks about how we're going to connect simulations that have different mathematical assumptions, which is exactly point number 4 out there, standards for describing the capabilities of, in our case, modeling frameworks, not just virtual tissue frameworks. But yeah, we need that language. We don't have it, but I think that's exactly what we propose to do. So, I would step forward and say, well, yeah, the Center for Reproducibility is going to try to do number 4 there. We'll see how far we get. But that's something that you write yourself.
Yeah. So. Well, so yeah, thanks for reminding me. But it, again, it feels like
I know it is one small part.
That's a bit. So maybe with the Center for Reproducibility, we can help with that. That 4 out of 5. Where do we start?
Number 4.
Yeah, we need the other ones. We need to coordinate so that we're not,
Yeah, I think your talk was great in that I think when we wrote the proposal, we were a little bit naive about not separating, as you talked about, modularization within a framework versus integration across different frameworks. And I think those are 2 important factors we need to think about.
And they're both important problems, but I think they are fundamentally different problems. I can have an operating system for a robot that runs on many different robots. But if I put two robots next to each other, they may not work together. And so they are different problems.
Less.
Yeah, so following up again on this, it's nice to hear about one aspect of the many challenges that you listed, and listed so beautifully. I mean, this is just really laying out all the work that needs to be done over many years. So it's nice to hear that one of them may be approachable, but you use the word hard in so many of your slides. And I'm wondering if you can distill for us, in addition to a particular project that we just heard about, what maybe some of the lower hanging fruit might be that maybe aren't so hard that we could address more easily. Have you thought about prioritizing in terms of what may be a little bit easier?
Well, I should apologize in the sense that I went through and tried to replace the word problem with challenge. And I should probably have taken the word hard out of the slide. I didn't want to discourage people. I do think that there are some of these problems in a sense are solved. Things like HLA architecture, which was developed for military agent-based application simulations, wargaming simulations, addresses some of these kinds of problems. The fact that there's a standard, however, doesn't help us necessarily because we have to implement it. We don't have the resources to implement them. I do think that developing translators, the way you're going to hear from Giuliano, is a good place to start. I do think that having the virtual tissue framework support common initial condition specifications, whether it's based on multi-cell DS or not, is something we can do. I think that agreeing that we're going to have certain kinds of interfaces and exposure within our simulation platforms is possible. I think that agreeing on languages at the conceptual level to specify components and their interfaces is possible. So actually, I think that there are a lot of these things that are useful even from the beginning. I also am a big believer in gradual standardization. And I think SBML has done this, which is the things that you do all the time that you agree on, that you codify as standards. And then you have a box that says, your arbitrary code goes here, and that's not standardized. That still has to be recoded. It has to be handled individually. Your hope is that with time, as you deal with more use cases, as you have more experience, the size of those your code goes here boxes gets smaller, but you always leave the option for extensions. You always leave the option for customization and breaking the standard. You can't enforce it rigidly the way you would, say, for cell phone interfaces, because there's scientific uncertainty. There was a comment yesterday about what happens if something we don't know about occurs. What happens if we discover something new scientifically? In those cases, we have to be flexible. And so I don't think it's realistic to ask for a standard that's universal and completely comprehensive, especially at the beginning, but to standardize the things that we can standardize. For example, chemical field descriptors and diffusion equation descriptors. Almost everybody solves those things in the same way and has the same choices. Those, I think, are easier places to start.
Yeah, Chicha?
James, I really enjoyed your talk. I have a question about the dynamic versus event-driven handler. I guess the time.
I'm sorry, that you're not picking up very well. Could I ask you to maybe to walk up to the front and ask the question closer to the computer so I can hear?
right.
I do appreciate people's patience with my not being there. I'd wanted to be there in person. I decided not to spread my bug uh, uh, to everyone.
Can you hear me now?
That's better. Thank you very much.
So I had a question about the dynamic versus event handler architectures, because that's kind of critical. So I'm just wondering, are you seeing them as one and the same? Because typically with some of these simulations, you're evolving time and then you're kind of checking on the states of the agents. And based on that, you can have an event occur. Or are you thinking about it in a different kind of asynchronous evolution?
Yes. So I had a slide which I think I flashed but didn't talk about, which is that I think that especially if you're thinking about interaction between software components, you have an issue, which is how do you do time and space granularity? Do you have a fixed time step? Do you have a fixed spatial granularity, the way CompuCell does, for example? Do you have adaptive time stepping, adaptive granularity? In the case of adaptive time stepping or granularity, are those the same everywhere in the simulation, or are they regionally variable? All of those questions really need to be addressed. If you want to do decomposition so that you're able to spread your computation out across multiple boxes, so you want to be able to do parallelization, not with a multi-core parallelization, which is typically pretty easy, but rather actually be able to discretize onto separate computers. Then you have to make decisions about when information is passed, how you deal with stale information, and so on. And so I think the question you bring up is a particularly good one. Typically, if you want to solve things really fast, you couple them tightly, but then you lose modularity. And so most of the solvers do just interleave time steps. You run some solvers for some short amount of time, then you'll do something like watch for changes, do your updates, run another solver. So you'll run the diffusion equation solvers, then you'll run the spatial evolution solvers, then you'll run the network solvers, and then you'll do the event watching at when you switch between solvers. And I think the best ways to do that, I think, are still out. It depends a lot on the timescales of the problem. And one of the things that we have to face, again, because we don't know how these things are going to be used, is that the same kind of architecture and discretization that's very efficient for one particular biological problem might be quite inefficient for another. As a simple example, when you do simulations of Dictyostelium discoideum and the movement of Dicty cells due to cyclic AMP relaying, if you build a very detailed model of cyclic AMP signaling, you discover that there's a very, very fast spike in the subcellular concentration. If you use a fixed time step for everything, then everything gets slaved to the fact that you need to have very, very high temporal resolution to get that spike to work. And so the simulations then become very, very expensive. But none of the mechanical properties of the movement of the cells operate on scales that are even 100 times anywhere near that. They're 100 times slower. And so in that case, some kind of adaptive time stepping would allow you to be much, much more efficient. You basically narrow your time window just during the spike, and then you spread it out elsewhere. Adaptive spatial discretization, remeshing, of course, if you do fancy meshing algorithms, auto meshing algorithms, say for diffusion solvers, why have almost everybody does diffusion on a fixed lattice, but why have lots of points where the concentration is effectively zero or isn't changing? On the other hand, if you go towards more finite element solids like modeling, where you do remeshing, then we have to rethink a lot of things. So I'm not trying to duck the question. I think it's a critical question to answer. But I do think if you're going to have modular components, you fundamentally have to give the module has to be able to run independently and then pass a set of flags to the other modules to tell them what to do, to tell them that events have occurred. And I do think dynamics and events have to be handled in a different way. So I think you've identified a critical point, yes.